In today's lecture, we are going to talk about basics of mass spectrometry, which could be used for characterizing, identifying the proteins of interest after performing the immunoprecipitation based experiment. So, in the last lecture, if you recall, we talked about if you want to identify the potential interactors of a protein of interest, you can do immunoprecipitation experiment and then identify the potential interactors using mass spectrometry. So, today we are going to have an application scientist who is going to talk to you about how to perform these experiments using mass spectrometers and then identify the possible proteins of interest using a software. So, let us have this lecture.
Hi, good morning everyone. So I am Pradeep from Thermo Fisher Scientific. I am the application specialist for mass spectrometry for proteomics and biopharma. So today, till now you have done the IP experiments. So immunoprecipitation, you do the antibody precipitation. So next, from those proteins which have been precipitated, how you identify the protein. Mainly we will focus on the mass spectrometry part. So my topic is mass spectrometry analysis from IP experiment to protein identification. So already you have done this experiment. So you have precipitated the protein and you have the protein. So what is the next step? Next step, either you start with this protein, you can run a SDS page, or in a solution you can digest the protein with some protease, which for example trypsin, which cuts after arginine and lysine. Then it makes a peptides mixture. So those peptide mixture is fractionated through LC and introduced to the mass spectrometry, where you get the masses of those peptides. And now you search against some database using some search tools, you identify those peptides. Okay. So today we'll discuss mostly on the mass spectrometry part, how you can analyze the data, how you can introduce the peptides into the mass, and how can you analyze the data. Okay, so before going to that, I am just giving you brief on the sample preparation. So you have the IP proteins you have. Either you can run a SDS page, normal SDS page, or you can make a solution in solution digestion. So in both cases, you in case of in-gel digestion, you run the gel in SDS page, then you cut the band of interest of your protein of interest, and cut the band, then you digest that band, extract. So when you keep with the trypsin, so protein get digested to peptides. So once it will be in a peptide form, it will come out from the gel bands, and you extract those peptides, and then further you proceed. So in case of in solution digestion, already there's protein in the solution. So you have to do the reduction and alkylation. It's very important part. Otherwise, what will happen? There are disulfide linkage. Due to this disulfide linkage, protein will not be open up. All the parts is not open up. So you have to go through this reduction and alkylation. DDT we are using is very common for reduction. It reduces the disulfides bond, and because of reversible reaction, you have to use some alkylation part. So block that sulfide residues. So you put iodoacetamide, and those sulfide will be blocked by iodoacetamide, and then you digest with some proteases. So most common protease is trypsin, which cuts after arginine and lysine. So the masses, basically important part is reduction and alkylation is very important, otherwise you will not get 100% digestion. Next, trypsin works in the basic pH. So when you do the digestion procedure, that has to be in the basic pH, more than 8 pH, okay, and it has to be kept at 30 degree. So these are the two criteria where trypsin works very well. And then trypsin, we all know trypsin cuts after arginine and lysine. Then after digestion, the cleanup procedure. So before injection to injected to the mass spectrometry, we have to clean up your samples. For in-gel digestion, it's fine, the samples comes pure. But in solution digestion, so samples may contain some impurities, some salts or some other impurities. So that will block your MS analysis. That's why the cleanup procedure is very important. So sample preparation that's why sample preparation is the most crucial part, and if you done a proper sample preparation, your 90% work is done. The next is basically MS is basically a software driven, so whatever you are set in the software, it will do, but the sample preparation is the most crucial part for yours. Okay. Now comes to the mass spectrometry. So I am keeping basics of the mass spectrometry, I am not going in very details on all the parts. So what is mass spectrometry? Mass spectrometry basically is a production of ions that subsequently filtered or separated by M by Z. Okay, so we are getting a resulting mass spectrum of abundance of produced ion as a function of M by Z. So any mass spectra, if you look into the mass spectra, so x-axis will be the M by Z value, mass by charge ratio, and y-axis will be the abundance, either relative abundance or the absolute abundance. Okay, so now sometimes if you run any samples in MALDI, okay, so MALDI top is very common. So that time sometimes you are saying your mass is coming. X-axis it comes as a mass, M. So whatever the M by Z value is the M, because in MALDI you are getting the +1 charge. So always the M by Z value will be equal to your mass. But in case of ESI, electrospray ionization, so that time you will get multiple charges. That time M by Z value will not be the exact mass. So don't be confused with this terminology. Okay, so now we can simply explain the mass spectrometry in 4 letters, GMSD. So it has, first is the generate, so you have to be ionize the molecule. Once any molecule can be ionized, it can be detected by mass spec. So that is a simple way to identify it. So it has to be ionized properly. The ionization method may be from solid to gaseous, maybe liquid to gaseous. Then it has to be moved properly. So after ionization till the detector, so that path should be the proper. Otherwise what will happen, if any molecule touch in the wall, that get discharged and it will not be detected in the mass spec. So that path should be very proper. Then selection, obviously you have to separate the molecule because you have a mixture of masses. So you have to filter which mass you want to identify, you can control that one. And lastly there is a detector. Okay, so you have to detect the molecule. So let's see what are the components are there in the different sectors. So first, ion source, ionization, where the ionization happens. So there are very different methods of ionization. It comes with the most popular is the ESI and MALDI. You already heard about this. Electrospray ionization, which is liquid to gaseous. So you inject the sample in the solution format, from where you put the high voltage, and that high voltage it will make the ionization. Next is the MALDI, matrix assisted laser desorption ionization. So it is a solid form to gaseous form. What happens in MALDI, you mix the sample with some matrix molecule, spot on a plate, keep it for dry, and then you shoot laser. That laser gives the energy to ionization. So from solid to gaseous phase. These are the two most soft ionization where your biomolecules will not degrade during the ionization. Whatever the other mode of ionization, fab, electron ionization, chemical ionization, these are very hard ionization, where already when you put your biomolecule, it will fragment in the source. So you will not get the exact mass of the molecule. So it is applicable only in case of small molecules like chemical compounds. And APCI and APPI, atmospheric pressure chemical ionization, these two are for non-polar molecules. Okay, exclusively for non-polar molecules. Now come for the mass analyzer, means where you separate your molecules. Most common, everybody knows a quadruple TOF instrument, time of flight. Quadruple means quadruple is not an analyzer, I can say it's a filter. It can precisely filter your molecule, which molecule you want. Ion traps, so you can put your molecules in the, you can trap your molecule inside a vessel and you can detect that molecule. So quadruple, ion trap, triple quartz, these are very low resolution. Means here you cannot separate nearby masses. But in the other case, FTICR, Orbitrap, TOF or QTOF, these are high resolution instruments where you can separate the nearby masses. Okay. So in IIT Bombay, we have Orbitrap technology. It's nothing but a trapping of electron in a orbital motion. Okay. So we put a high voltage and the ions are rotating around the molecule. Okay. So we'll come to those parts. And last is a detector. So you require some detector. The better part for Orbitrap is Orbitrap itself acts as an analyzer as well as detector. So you don't require an extra detector for Orbitrap. Okay. Now we see how the mass spec works, what we are doing in the mass spec after injection of sample. So we inject a sample to mass spec. Through the mass analyzer and detector, we get the MS1. Okay, MS spectra. We'll get this MS spectra. From this MS spectra, we will select one of the peak, and again we will do the fragmentation, MS2, and we get this fragmentation. Now this fragmentation will search against the database, and we will find out what are the ions are there, B and Y ions, and we get the database match. We will get the peptides. From the peptides, we can say this protein is present in your sample. Okay. So now come to the nomenclature of identifying the ions. This is basically nomenclature how you can assign your peptides. So this is example of a peptide of 4 amino acid R1, R2, R3, R4. Okay. So now when you applied voltage to fragment the molecule in MS2, so this C alpha C, Cn and Nc alpha, these 3 bonds can be broken. So this any one of the broken. Okay. So if it is C alpha C bond is broken, then the N terminal side will call the A ions, the C terminal side will call the X ion. Same way Cn, this is the peptide bond basically, B and Y ions, and if it is Nc alpha bond, then it will call the Cz ions. Okay. So now there are different kind of fragmentation, different way of fragmentation, CID, ACD, ETD. So different fragmentation will give you different kind of ions. So in case of CID, you will get only B and Y ions. So in case of ETD, you will get C and Z ions. Okay. Now come to the facility here in IIT Bombay. So it has a tribrid instrument, Orbitrap fusion, coupled with nano LC. So why we require nano LC? Because we are doing the proteomics. In proteomics, sample is very small amount and it is very precious. So we cannot run the normal HPLC where the flow rate is very high. We have to run in the nano LC in a nanoliter flow rate. Okay. Now come to the schematic diagram of Orbitrap fusion. So Orbitrap fusion, as I told, it's a tribrid instrument. It has quadruple, it has ion trap, and it has Orbitrap. Okay. So ions are inserted from here. It gets filtered in the quadruple first. Then it comes to the ion routing multiple where the ion will store, and it will tells you where to go because you have two detectors, ion trap and the Orbitrap. So from here either it will go to the ion trap or it will go to the Orbitrap. So then Orbitrap basically is a high resolution. It gives you 500K maximum resolution. And lastly the ion trap, which is basically a dual cell ion trap. You can store your ion and you can do the detection of ions. And also you have a ETD option. So in the next slide it will be clear for you. We have a video how it works. Now we are doing first the full MS1. Okay, so now ions will generate here. It will passes through ion transfer capillary, inserting through that. So after that, here what happened, ions trying to spread out because all the ions are very highly charged. So it tries to spreading. So you need a focusing. So it will focus. Then it's called the bent flutter pole, benting, where the neutrals are removed. Because when the ions are inserting to the instrument, there are some neutrals also. So you can, with the high voltage difference, the charge molecule can bend, but the neutrals cannot bend. So after that, in the quadruple, you can select the ions, which one you want. Then it first comes to this ion routing multiple, where it will first store here. So here is the first stoppage where it will be decided either it will go to the orbit trap or it will go to the ion trap. Okay. So now it will come to the orbit trap, MS, and then it's been detected in the orbit trap. Okay. So now it's a full scan. What happened in the MSMS? So in case of MSMS, only one molecule has been filtered from this mass. Only one molecule filtered, and that gets fragmented, and then it will be detected, MS2. So same thing happened, ions are going. So inside a quadruple, the only one molecule will be there, so other will be filtered out. Now that molecule go to this ion routing multiple and get fragmented. So here it will fragmented in the ACD mode. It's nothing but the collision induced fusion with high energy. So here after fragmenting here, it goes to the ion trap, where it is stored first and then trapped here, and then it's get detected. Okay, so it's detected here with the large surface area detector. So it's a very parallel reaction, basically parallelly it is going on. So when the first MS is going on here, MS1, at the same time the MSMS is going on simultaneously. So it's a parallelly both the detector is working. Okay, so from one MS scan, it can do 20 MSMS. It can select 20 peaks from one MS scan, and it can do the MSMS of 20. Okay, now come to the specification of fusion orbit trap. So first is the mass accuracy. Mass accuracy is tells that is less than 1 ppm and for internal calibration, and with less than 3 ppm with external calibration. So what does it mean by external and internal? So every instrument requires a calibration. So you have a standards, you inject the standard. So you have the mass of the standards, and against that masses, exact mass, you calibrate your instrument. So that is the simple external calibration. Now in case of internal calibration, we fix one of the masses as a internal standard. We fix that mass and do the again calibration. So that in that time we will get the internal calibration. So that's why when we do the internal calibration, the calibration will be less than 1 ppm. Now the mass range, so you can acquire the mass range from 50 to 6000 m by z, not the mass. Okay, so m by z. So bigger the molecule, the more the charge state, and it will come inside this range. But you cannot scan the whole total mass range 50 to 6000 in one shot. So you can scan the first mass, and the highest is the 15x of the first mass. So for example, if you start from 100 m by z, you can scan up to 1500 m by z. So if you want to scan the full mass range, then you want to divide the scan range. So start from 100 to 1500 and 1500 to 6000. Then resolution, you can get the maximum resolution of 500K at 200 m by z. Scan speed 18 hertz per minute. So fragmentation, so you have three different kind of fragmentation: CID, ACD, and ETD. And there is a basically mixture combination of any two, ETD, ACD, and ETD, CID. So in case of CID, it's basically collision induced dissociation. So what happened in that collision induced dissociation? You collide your you inject some neutral gases that will collide with your sample molecule and get fragmented. That is CID. ACD is high energy collision induced dissociation. So this is nothing but CID with higher energy. We are putting higher energy to get better fragmentation. And ETD is a electron transfer dissociation. What happened there? You put a reagent molecule which ejects an anion. That anion will collide with the sample molecule and get fragmented. That is called the ETD. So according to your sample objective, what do you want to do, according to you have to select the fragmentation mode. Then polarity switching means it's not required for the proteins, but in case of small molecules, if you don't know in which mode, either it's a positive mode or a negative mode, it will ionize. So you can do the both scan, positive scan and the negative scan within a 1 second. So you can run that one way. MSN, as it is a ion trap, so you can do 10 MSN, means MS2, MS3, MS4, up to 10 theoretically. Isolation in quadruple, you can go minimum 0.4 AMU, means plus minus 0.2 Daltons. So what that means if you want to identify one molecular M by Z of 100, so you can scan between 99.8 to 100.2. So that narrow window you can scan. Okay, so now comes see some experiments where means how the instrument works in different workflows, because we cannot show you in the instrument part. So we have some animation where we can see how the instrument do the MS and MSMS. So first experiment tells ke MS in OT, orbit trap, and MSMS doing the through the CID, it will do in the ion trap. So CID will be happened here. So what happened, first the ions will be detected here, MS. From that MS scan, one molecule one at a time, it will be filtered here and fragmented here. Okay, so let's say, so the first full MS will go. So the first mass, it will stop here, then it will comes to orbit trap. When the full MS is going on, we are doing the parallel. So the second first parent ion has been selected here. Okay, and it will be stopped there first. Then it will go for the fragmentation in the HPC cell. When it is going fragmenting, the second ion is waiting here. So you are not losing any molecule as well as the time. Okay, so it's a parallel reaction. So when all the MS2 scan is over, then only you will get the MS scan. So at the end of the full scan, you will get MS scan as well as MSMS scan. So you are not losing any time as well as any molecules, samples. So like second experiment is the ACD. On the first experiment we are doing in the CID. What happened? In that case, the fragmentation happens here. Now in case of ACD, the fragmentation will happen here. Okay. So now same way, the full MS, it will stop here, then it will go to orbit trap for analyzing. Now the first mass will comes here and get fragmented. When it will be sending to the ion trap, at the same time the second mass will come up. So you are not the same way, you are doing the parallel reactions. So after all the MS2 scan, you will get the MSMS scan. Now next is the ETD. What happens in case of ETD? So even the ETD fragmentation will happen in the ion trap also. So first is the full MS. In same way, the full MS comes to orbit trap. Now the first ion has been isolated in the quadruple and first stop here, then it will injected to the ion trap. Now I told that in ETD you require a reagent which gives you the anion. So that reagent will comes from here. So it will inject a reagent. Its reagent calls the fluoranthin. So that is a carcinogenic. So it will gives you some anion. It will make the anions and get fragmented that molecules. So once the fragmentation is happened, so it will give you the MS2 data as well as MSMS data. So ETD is a very specialized case only when you are going for the PTM type analysis. So post-translational modification like phosphorylation or acetylation or glycosylation. In those cases, you have to use this ETD reaction. So in the instrument, the better part is that you already there are already the templates for all type of analysis. So you don't have to start from the scratch. So you just click on the which method you want to use, either it's identification or it's a quantification or is a PTM work. So you have to select that method and run the method. Okay, so there are already templates are made. Now before going to the next step, so LC is another big part, because in the front end we are putting a LC. Why we require a LC for this kind of analysis? Because the peptide length may be from 300 to 2000 m by z. So within the mass we cannot separate all the m by z's. We can separate the nearby masses by the resolution, but we cannot separate in the range. So we need to separate those peptides. So that we put a LC in the front line, where either with the basis of hydrophobicity or any other parameters, we separate those peptides and elute one by one and inject it to the mass spec. Okay, so these are the different parameters we are using for setting up the LC run. Here the important part is the gradient. So you have to choose a proper gradient. So that gradient should be on the complexity of a sample. So now here I am showing you 30 minutes gradient. So it's work for a simple one protein like BSA or single protein. But when your complexity, when you are working with the wholesale lysate, that time you have to increase the gradient for 2 hours, 3 hours, 4 hours. Okay. So like that you have to work on the gradient part, not in the MS part. Okay. So now come for some data analysis part. For data analysis, we have Protein Discoverer 2.2. That is a comprehensive and extendable software for qualitative and quantitative. So you can identify as well as quantify the proteins. Here the quantification is a relative quantification, not an absolute quantification. Okay, so so what it has, basically it will have some different search engines like Sequest, Mascot, Bionic, Amanda. Out of this, Sequest is a free available, so it will comes with the integrated with the Protein Discoverer. Other search engines, Mascot or Bionic, those has to be purchased with the license. So actually what this search engines works, means how it works. So when you have run a sample, human sample, you get the data, MSMS data. So now you tell the software search engine ke you extract the protein, you set the database of human. So the search engine will theoretically digest those protein from the human and make a theoretical mass list. And then the theoretical mass list will be compared with the experimental mass list. At the MS level, first MS1 level, when it will match with the MS level, then it will come for the MS2 level. If it is match in the MS2 level and get those B and Y ions to get the sequence, then it will tells you, okay, that peptide is present. Then it will give you, okay, this protein peptide is present, means this protein also be present there. Okay. So it will determine relative quantification. So you can do relative quantification, either it's a label free or a labeled reaction like TMT, SILAC, ITRAC. So here is a window where you will see the first off is a wholesale lysate run for 120 minutes. We run a DDMS2 method. We do OT at MS and ACD also in orbit trap. Okay, MS2 also detected in orbit trap. We are not using ion trap here. It's a 180 minutes run. It's a wholesale lysate. So first window, it tells the TIC, total ion chromatogram. So like in HPLC, you are getting UV chromatogram. Here we'll get on the basis of total ions, it will make a chromatogram. So that's why it's called the total ion chromatogram. Second window is called the BPC, base peak chromatogram, on the basis of highest intense speed for the each retention time. Okay, so that is called the base peak chromatogram. Now if you click anywhere, you will get like this of MS spectra. This is the real MS spectra, where x-axis is the m by z, y-axis is the relative abundance, and it will see different charge states are there, +2, +3, +4. Because of ESI, you will get the different charge states. Okay. Now when we search this data in PD with these two workflow. So in PD you have two workflows. One is the processing workflow, where we search the data against the database using the Sequest, and after getting the peptides, those peptides may be a false positive or false negative. So you have to validate those peptides. So those has been done in the consensus step, where we do the peptide validation as well as protein validations. and we will ultimately get the data. So here the string we are doing the validation at 1% FDR. So what does that mean? If you run the sample 99 times, the protein will identify it. Maybe in one time it not been identified. So in such stringency we are detecting validating those peptide, and we are telling, okay, this peptide is present. So then the report is very good for publishing. So now after search the data, what is the report format? So it comes like this. So these are giving you the protein information, what are the protein match, and the sequence coverage, how many peptides has been matched. So here important thing is the unique peptides. So we are looking for the unique peptides. How many unique peptides we have identified? So what does mean by unique peptide? Unique peptide is those peptides which are present only in those proteins, not any other protein included in the database. So what database you are using, in that database that peptide is not present in any other protein. So we are looking for at least 2 or more unique peptides. Those kind of data will be the very good data. Okay. So here we have identified almost 2255 protein groups. So it comes like from that data set, it has extracted this many MSMS spectra, which is when we match against the database, we are getting this many 23,000 PSMs, and out of these PSMs we are getting this many 2255 protein groups. So right now I'm talking about the identification, just how to do. So fusion is not for identification, it can do many works. So right now we are doing the deep large analysis and the label free or label reaction to relative quantification. So it can do the phosphoproteomics, it can do the lipidomics, it can do the metabolomics. So you can use this instrument for different kind of analysis. Lastly, this is a website planetorbittrap.com, where you will get all type of information of Orbitrap in regarding application wise. If you are working on proteomics, so you will get all type of application notes, published data or methods. So you can register here and you will get the free updates. Okay.
So from the last 2 lectures, you are now familiar that if you have a protein of interest which you want to dig deeper, you want to further characterize that protein and want to understand the possible role of that protein, an easy way of moving forward could be to do immunoprecipitation experiment followed by protein identification using mass spectrometry. In today's lecture, you have seen how to take the complex which you have eluted out from the IP experiment and identify the potential interactors using mass spectrometry. There could be many softwares which could identify the potential proteins of interest. In fact, I would recommend you to use one of the open access software, Mascot, to do the data analysis, where you can easily identify the proteins of interest. There are many good softwares available, even commercially. One which we have shown today is Proteome Discoverer. In general, I hope these 2 lectures have given you some basics and understanding about how to study the biomolecular interactions using immunoprecipitation followed by mass spectrometry based experiment. Thank you.